We will talk today about the cytoskeleton. We will just imagine ourselves or our bodies without the skeleton itself, the bone skeleton. How would it be? It will not be rigid. It will not be straight. There will not be a specific shape for it, and it's the same in the cell. Also, the cytoskeleton will shape out the entire cell, and it will give it the rigid or the specific shape it should be in. What's the objectives for today? We will know the definition of the cytoskeleton. We will know the function of it, and we will describe the structural component of it, and then we will describe the structure and types of filaments and functions of each of these filaments or types. There are two main roles for the cytoskeleton. First of all, it consists of a network of protein filaments extending throughout the cytoplasm. There are two roles, as we said. There is a structural role, which providing a framework for the cell that determines cell shape and position of the organelles. Each organelles will positions in its specific place or location. And there is also a transport role, which is the cell movement and transport of organelles through the cytoplasm. From its name, you can imagine that it's rigid as the bone skeleton that we have, and it doesn't move. But in real, it's a dynamic structure that is continually reorganized as cell moves and change shape. So it's a complex mixture of different types of proteins responsible for providing this mechanical strength to the cell and support the movement of cellular contents. As you can see here, it consists of three types of filaments. There is what's called actin filament, microtubule, and intermediate filament, which is in between the actin and microtubule. That's why it's named intermediate filaments. All are held and linked together to subcellular organelles, as you can see here, and plasma membrane by a variety of accessory proteins that we will cover later on, and we will get through it later on. First of all, let's talk about the actin filaments. They are considered as the most abundant skeletal protein. They consist of what's called actin that will polymerize to form actin filaments. As you see, thin, flexible. It's 7 nanometer in diameter and up to several micrometer in length. It's called also microfilament. There are two names for this actin filament. We can call it also microfilaments. It's organized into higher order structure to form a bundle of 3D network with properties of semi-solid gels. It's regulated by actin binding proteins which are responsible for the assembly, disassembly of actin filaments, cross-linking into bundles and network, as we will cover later on, the association with other cell structure. So these are the main characters or features for the actin filaments. They enable cell also to function. They make cell able to migrate, divide, sorry, and engulf some particles, and it allow also the movement of surface. It's abundant beneath the plasma membrane. As we said, it will form like a network to provide the mechanical support. It also will determine the cell shape. It will allow the movement of cell surface. It will enable cell to migrate, engulf particles, and divide, as we said. Some are presented in nucleus too, mainly responsible for large-scale changes in cell shape during cell movement or contractile ring formation. Here you can see how this actin filaments polymerize. Um, the the actin molecule is a globular protein. It's around 375 amino acid. So these the G-actin, which are the actin molecule. They have a high tight binding site that mediate head-to-tail interactions with other monomers. So they will polymerize and form filaments, which are the F-actin or filamentous actin. All are oriented in the same direction, have distinct polarity. The ends called plus and minus. As you can see here, the G-actin is the subunit. It will form like a nuclei in a process called nucleation, where the building of the other or the elongation of the full filament will begin. So the elongation here will happen with the addition of the G-actin subunits and dissociation from the minus side. These plus and minus are not charged, they are the sides or the place where the actin will assemble or deassemble. If it's deassembling, it will be minus, and when it assemble, it will be plus. It's also we can call it a pointed or barbed end. So the pointed will be the the plus, while the barbed will be the minus. Sorry, yes, the opposite. The barbed will be the plus, and the pointed will be the minus side. So here you can see from this animation how it's nucleate here. Then the addition will be on the plus end, and the association and disassociation of these actin will just continue to perform what's called a trade milling, where the rate of addition and rate of association is the same. So when the rate of dissociation, deassembly, and assembly are the same, then it's called the trade milling. In the barbed end or in the positive plus end, it grows 5 to 10 times faster than the pointed end, which is the minus. And you can see here that it needs ATP. It acts as a ATPase, where binds ATP hydrolyzed to ADP. So once there is ATP, it will bind, and once it's lose the phosphate, the P phosphate, and it will be the ADP. So it will be disassociated from the filament. You can just watch these videos for more clarification of the assembly of the microfilaments or the actin filaments. To initiate the actin lengthening or shortening, there are several regulations for this microfilament. There are what's called actin binding proteins that will modulate the assembly or disassembly of this actin microfilaments or actin filaments. There are several types of proteins. We can summarize it as these in these 5 types. We have the proteins, the actin binding proteins that will increase or will associate to the nucleation rate. As you said, the nucleation is the formation of the nuclei that will be the start point to build up the filaments. Here the protein is called ARP, which is actin related protein 2,3, that will increase the nucleation rate and will just get this microfilament. As you can see, the minus and plus, the plus and the minus ends. When this ARP protein associate to this actin filament, it will cause the branching here. So this will lead to formation of branches. There is also what's called plus end assembly proteins, which are here, for example, the profilin, which associate to the plus end and will increase the rate of addition or assembly, and then it will lead to the lengthen or increase the length of this filament. We have what's called a cap Z. This cap Z will associate or will attach to the plus end, and it will block the addition. So if the plus side is capped or blocked, then by default it will not be any, there will not be any addition, so the disassembly will be more here. So this filament will be shortened, it will lose these G actins. We have also the minus end protein, binding protein, which will it's the same, it will block the disassembly. So it will free out the plus end, which will mean that the G actins will bind, and it will be more lengthy here, the filament. We have also the stability of this microfilament by the addition or the attachment of the tropomyosin, for example, and this will cause it will just attach to the sides and will cause the stability of this filament, and you will find it in many type of cells. We have also severing or cutting proteins like the cofilin. The cofilin when it binds to this microfilament, it will just lead to the cutting of this microfilament. So these type of proteins are essential for the cell division and the motility of the cell. There are some type of cells that contains many type of these proteins, like the muscle cell. It will have the capsid, for example. It will have the thromboglobulin, and it will also have the tropomyosin. So these are the proteins that regulate the length of the microfilament, when it's long and when it should be for example shortened, when to be divided, and where to be and when to be actually branched. These are very essential in the formation of the microfilaments. The microfilaments or the actin filaments can be assembled into main two types, actin bundles and actin network. The bundles, as you can see here, are cross-linked into closely packed parallel arrays. Here you can see two parallel filaments are linked to each other by this type of linking protein, which is here the fibrin and alpha-actinin. So these are the bundles, while the network are cross-linked in arrays to form 3D meshwork. So here will be packed parallelly, and here it will be just cross-linked in arrays to form the 3D meshwork. The actin bundles will be cross-linked by actin bundling protein, and these proteins has two domains to bind actin and align the filaments, as you can see here. There are two types. There is what's called non-contractile or parallel bundles, and there is a contractile bundle. The parallel bundle or the non-contractile will be around 14 nanometer apart from each other. They will align, as you can see, in parallel, same polarity, barbered ends adjacent to plasma membrane, and this protein, the fibrin, will just cross-link each filament with the other with two actin binding proteins, so binding domains. So this type of protein has two actin binding domains, so it can bind on both sides, as you can see here. That's why it's parallel. So this type of bundles are responsible for the cell movement, phagocytosis, absorption of nutrient, intestinal microvilli, membrane projection to increase absorption of the surface by the, as we said, the microvilli. While the contractile bundles are 40 nanometer apart. So here it's 14 nanometer, while here it will be 40 nanometer. So it will be more space between the two filaments. It will be cross-linked by alpha-actinin, as you can see here, which has a single actin binding domain. So here in red you can see each protein or each actinin has single actin binding domain. This will interact with actin as dimers, as you can see here. They will be as dimers and will attach to each filament. This will increase the spacing and will allow actin interaction with motor protein as we are going to see later on, which is the myosin, and these are responsible for some muscle fiber contraction. So these are the bundles that assembles as actin filaments, while the actin network, as you can see here, there is what's called the filamen, which form a flexible crosslinks. These are considered as dimers, as you can see here. They are a flexible V-shaped molecule. They have actin binding domain, as you can see here in red, at the end of the arm, while they have the beta sheet spacer domain, which is this one, and they will just dimer, and there is also the dimerization domain, and they will bind the they will cross-link the filaments in like a 3D shape, mesh shape, and they are located beneath the plasma membrane. They will determine the cell shape and the movement. There are what's called motor proteins. These motor proteins will bring higher level of functional complexity to the cells. We will find them in cellular movement, intracellular transport, and cell division, for example. One type of these proteins are the myosin. They are considered as a molecular motor that converts the ATP, the chemical energy, to mechanical energy that will lead to the force and movement of the cell. You can find them especially in the muscle fibers, which will be together with the actin filament, which will lead to the building up of the muscle. As you can see here, the myosin is a large protein. It has a 2 heavy chain and 2 pairs of light chain. It has a globular head that will bind to the actin, and there is also the motor domain, and we have the regulatory domain here, and we have the tail domain. It's alpha helical. The tail is alpha helical, and it just consists of, as we said, 3 domains. Each myosin has 3 domains. The motor domain will bind to the actin. As you can see here, the myosin will attach to the actin microfilament. Phosphate will be released. Head will slide the filament, as you can see here. The ADP will be released. A new ATP attached to the myosin head, as you can see here. It will link between a link between the myosin and actins weaken, and this will lead to the detachment of this actin, and ATP will split into ADP and ATP. Will be ADP and phosphate. Then the myosin head will be energized, which means it will have a high energy conformation, and then it will attach to the actin. So it's like a cycle, depending on the ATP, the hydrolysis of ATP. Once it's hydrolyzed, then it will just detach to this actin. So this is how it moves, and the attachment and detachment of this myosin will lead to the movement of the actin filament. The next type of the filaments are the intermediate filaments. As you can see, they have great strength. They enable cell to withstand the mechanical stress associated with stretching. It's approximately 10 nanometer in diameter, and it's considered as the toughest and most durable. It's found in cytoplasm surrounding nucleus, extending out into the cytoplasm. It's found in tight junctions in nucleus to strengthen the nuclear envelope. As you can see here, it consists of C-terminal and N-terminal. There is head and tail. The C is the tail, while the head is the N. And there is alpha helix, central rod domain for each subunit. And this is like a globular shape. They will form dimers, so each, it's like a rope. You know, if you have a thick rope and you just untwist it, you will find out that it's reassembled in this exactly the same shape. So one the one fiber or one filament will bind or will wind to another one. So two filaments will cause the dimer to present. Then there is what's called a tetramer, where two dimers will attach, so it will cause four filaments to be attached, and so on. So until it causes what's a 13 here, a 13. Once you cut it, you can see that there is a 13 circle or 13 filament. This will cause the building up of this intermediate filament. As you can see here, the head, tail, as we said, and the rod domain, there are just a few types of these intermediate filaments. You can see here the keratin. There is acidic keratin and there is basic keratin. Acidic keratin and neutral basic keratin. You can find them in the epithelials. Here, the epithelial, and here's also the epithelial or the epidermal keratins. These hair shaft and nail, they all consist of this type of intermediate filament. And there is also here the vimentin, which is making up the fibroblast and the blood vessels. For example, there is also the neuro filaments here, which makes up the nervous system or the neurons. There is also the nuclear lamin or the nuclear lamina, as you can see here. It makes up the nucleus, and there is also the nestin that makes up the stem cells. These are the type of the filaments or the proteins that make up the cells that are, as you can see, they are essential for the human body. Very important to make up the organs. The last type is the microtubules. The microtubules are long, stiff, hollow tubes. They tend to grow from central locations within the cell, which are the centrosome. They create a system of track across cells. Physical organelles travel like trains along these tracks, so they keep the organelles in their location. They reform as mitotic spindles during cell division, and they will form the core of cilia and flagella that we talked about before. They consist of alpha tubulin and beta tubulin, here in green and blue colors. These tubulins can grow in a certain rate, but to increase this rate, there is what's called the gamma tubulin. The gamma tubulin will form the centrosome, as you can see here. This is the centrosome, that these microtubules will start to build up. So it's like a center, the centrosome, and these microtubules will be built up and just grow out from these centrosomes. So as we said, the microtubules are formed by outgrowth from the specialized organizing center, here the centrosome, present on one side of nucleus. Its complex structure composed of gamma tubulin, here in yellow, subunits. The alpha and beta subunits grow from them. So once you can see here, the dimers the alpha and beta will form what's called a dimer here. So then they will keep attaching to each other. Once there is a ring with 13 protofilament, it will build up. This nucleation step will build up other rings and so on to form this type of like a tube or channel, as we said before, for the molecules to just pass by. There is what's called a dynamic instability. To build up these tubulins, it uses the ATP to grow and contract. So the the material will across their length by motor proteins. As you can see here, there are several types. They differ only in the cargo they bind and the direction they travel. For example, there is here the kinesin and the dynein. You can see here the kinesin will just move toward the minus end, while here the minus end, the dynein will move to the minus end, while the kinesin will move toward the positive end. So they are like walking on opposite side. The kinesin, as we said, will move toward the plus end, while the dynein will move toward the minus end. As you can see, there are domains to bind with the microtubules, and they will compose of multiple subunits and chains, and they will just hold the molecules or the substance that has to move through this microtubulins and toward the specific place it has to be cargo to it. So this is the main things about the cytoskeleton to focus on, and to know about. You can just read more in this book, the Cooper, and watch the videos for the assembly of the actin filaments.